Okay, so good morning. I hope everybody had a really nice spring break. Mine was really nice. It was just way too short, right, which I'm sure is what all of you probably feel also. Um, so let's get back into our discussions about different considerations that influence the development of cancer. Um, to start off with, I wanted to just do a quick reminder. Here is our schedule. Here we are in week 10, so we are at lecture 21. Today we're going to talk about um, considerations of organism size and lifespan in terms of being evolutionary considerations for how to deal with cancer. Then on Wednesday, we'll talk about the heterogeneity of tumors, talk a little bit more about DNA repair mechanisms. And then Friday is a university holiday, right, because it's Kuhio Day. Next week, Monday, we'll have new material as well. We'll talk about sources of DNA damage and different mechanisms that are used by our cells and tissues for actually resisting cancer. But here is the sort of, hopefully this is not a big surprise to all of you. Next Wednesday is our second exam, right? So unfortunately, because of the 2 short weeks, this week Friday being Kuhio Day, and then next week Friday being Good Friday, it sort of ends up compressing our schedule a little bit through these couple of weeks. So we won't have a day of simply reviewing the exam material. That's why it's very important that coming off of spring break right now, you're already aware that this weekend, coming up as a long weekend, you should definitely be studying. And then on Monday, we will talk about some new material, and you will be responsible for that material on the exam next Wednesday. Okay. In addition, on the right, there's a reminder that you will have a quiz that's due this week, Wednesday, normal time, and that will be on chapters 10 and 11. And then next week, to help you as part of studying, there will be a homework assignment, and then you will also have a quiz. Okay. So keep those in mind as we head through this week. Let's review where we left off to make sure we're all on the same page again. We had been talking about telomeres, and we said that telomeres are these repeats of TTA-GGG that repeat at the end of our chromosomes. And I had likened these telomere repeats to being sort of like the free end pages that you have at the beginning and the end of books, right? Because these are pages in the books that can be torn out, and you don't end up losing any of the actual content of the book itself. Likewise, our chromosomes are designed that way. We have these many kilobases of repeats at the ends, and these chromosomal regions serve several important functions. They protect the genome from chromosomal damage, right? So that you can actually lose bits of your ends of your chromosomes before you actually get into potentially losing valuable genetic information that's in the chromosome. The telomeres provide a physical platform for DNA replication to take place. Right? Remember, DNA replication requires the use of Okazaki fragments on the lagging strand as you're replicating your DNA. So with every cell division, you're going to have random RNA primers that need to settle down at random spots on the lagging strand to make the short Okazaki fragments that represent, the copy of the DNA on that side of the replication bubble, right? And we said that it's very unlikely to imagine that you'd have RNA primers that perfectly line up with the very end of the molecule of DNA, right? The very end of a chromosome. It's way more likely that somewhere further in would be the last RNA primer that actually successfully starts copying. So you would lose that bit of information at the end. So our telomeres are very useful for that purpose. Right? But as a result, with every cell replication, right, the telomeres get shorter and shorter. And as far as our fully differentiated cells are concerned, that's a very useful way of keeping track. It's like a tally, right, of how many cell divisions a differentiated cell has gone through. And once the telomeres get too short, we said that that cell enters what we call crisis, right? And that's when it needs to stop replicating. And crisis may go hand in hand with senescence, right? Where the cell actually stops doing its normal functions. And this is very much the case for the multitude of different fully differentiated epithelial cells that we have in our body that maybe only last for a week or so, right, before they're just lost, right? So they may go through crisis, they may go through senescence, or they might just be physically lost as part of their normal function. So we said this is important to understand because in order for a cell to transform and go all the way through transforming steps to become full-blown cancer, then one of the critical things is that this transforming cell has to overcome crisis and it has to overcome senescence, right? So these things need to be overcome in order for that cell to become what we think of as immortal. So here's the thought question then. Very clearly, as we've learned, these telomeres at the ends of our chromosomes play a very important role, right, in being able to maintain or keep track of things like genomic integrity, how many replications a cell has undergone, right? And the telomere lengths then play a very important role in triggering things such as crisis and senescence, right, as a mechanism to stop our fully differentiated cells from being immortal, right? So if the telomeres are that important in our cells, very clearly they must be important in other animals as well, right? So let's think about this mouse here, for example, right? Do telomeres in mice behave the same way, right, as telomeres in humans? You know, on the one hand, you might not even think about that because you might think, well, yeah, I mean, we're mammals, right? So obviously we're very similar evolutionarily. And so clearly, right, the mouse genome must depend upon telomeres the same way that we do, right? But is that necessarily that obviously true? Okay, let's look at this. If you look at the typical body size of a mouse, right? A typical mouse, let's say, is 40 grams, right? Compared to a human. A human is about, on average, 60, 70 kilograms, right? So that means an adult human is about 2,000 times, right, bigger body size than a little mouse. Let's think about lifespan. A typical mouse, in the wild at least, might be expected to live for what, maybe 1 to 3 years. A typical human these days, we can easily expect to live 70 to 90 years or even more. That means the human lifespan is about 30 times longer than the typical lifespan of a mouse. Right? So let's think about these two things, body size, lifespan, and let's take a look at the size of telomeres. A mouse's telomere is typically about 40 kilobases of telomeric repeats. This is compared to human telomeres, which are about 10 kilobases. So interestingly, right, even though we are way bigger, 2,000 folds larger body size, and we live like 30 times longer than the mouse's telomeres are about 4 times bigger than human telomeres. So that's no accident, right? Very clearly, evolutionarily, something is happening here, right? That doesn't seem very intuitive at first glance. But the key is thinking about body size and lifespan. Because body size, you can roughly think about. That means how many cells are in the body, right? And each one of those cells, if it's a nucleated cell, has the potential to transform and become cancer. Right? So you might say, conservatively, we have about 2,000 times more cells that potentially could become cancerous in our lifetime compared to a mouse. Right? And lifespan, because the longer you are alive, that literally is more time to accrue or build up mutations in your genome, right? So if you think about these things together, what are we dealing with? We are dealing with the fact that in our bodies, it is far more likely, right, to develop transforming cells that are on the path to becoming cancerous because we have more cells to start with and we stick around for way longer. So why then would the mouse's telomeres be so much larger? Right? So this is kind of a critical thinking thing here, right? If you think about the functions of the telomeres, we said telomeres first and primarily, they protect the ends of the chromosomes from sticking together, right? Otherwise, without the protection of telomeres, the chromosomes in your genome might end up joining end to end, and that'd be really bad, right? Telomeres provide a necessary platform to be able to replicate DNA, and in the process of doing so, minimizing the risk of losing valuable information. And number three, right, telomeres also serve as a clock for cell divisions. So if you don't have nearly as many cells to worry about transforming, and your lifespan is short enough that you're not really worried about accruing mutations that turn into cancer, right, then maybe the way you evolve telomeres is to make sure you have really big telomeres so that you are really protecting your chromosomes from sticking together, and you're making sure that you have plenty of telomeres to replicate your DNA accurately. Right? So therefore, maybe that's why mice have larger telomeres than humans. Well, let's use a different example. Maybe something, at the opposite end of the spectrum. Mice are teeny tiny compared to people. What about other mammals that are way bigger than people? Right, let's look at an elephant, for example. So an elephant's body size, a typical elephant, many different, several species of elephants, but typical body size might be 5,400 kilograms. Again, compared to the typical 70 kilograms of an adult human. So an elephant is 77-fold larger, right? Larger body size than a person. Let's look at the lifespan, right? An elephant lives for about 50 to 70 years. We live 70 to 90 years. So it's pretty similar, right? So the lifespan of the elephant's about the same as a human. That means a similar length of time to build up potentially deleterious, bad, transforming mutations in the genome. Right? The body size of an elephant, though, is 77 times larger, right? So that's way more cells that potentially could transform. Right? And yet, look at the telomere length, right? An elephant's telomere length is 15 kb compared to 10 kb in humans. So it's very similar. So again, this is an interesting observation, right? So whereas in the mouse, we said, well, the mouse is small and it doesn't live very long, so therefore it's not as concerned evolutionarily about cancer, so it can afford to have really big telomeres because it doesn't care as much about keeping track of differentiated cell division, right? An elephant, on the other hand, lives about as long as we do, has way more cells to worry about that could potentially transform, and yet its telomeres, right, aren't that much bigger. So how does something like an elephant deal with preventing cancer? How does the elephant successfully manage or maintain some sort of careful watch over all of the cells in its body to make sure that you don't have unwanted oncogenic activity, right? How has the elephant evolved to cope with having many more cells and therefore many more opportunities for mutations? Well, the answer, I think, is really, really eye-opening, right? Because if you're going to have that many cells to worry about, right, and you're worried about undesirable mutations that might cause oncogenic mutations, right, or oncogenic changes in the cells, you really want to make sure what types of proteins are really active. You want to make sure that your tumor suppressor genes, right, are really, really good to be able to catch this. How does the elephant make sure that it has good tumor repressor functions? Because the elephant's genome has 20 copies of the p53 gene. Right? compared to other mammals like us. We just have one copy, right? The human genome has one copy of p53. So you have one copy from your dad and one copy from your mom, right? But in the genome itself, right, each version of the genome you got from your parents, there's one copy of p53. An elephant's genome, right, has 20 copies from its mom, 20 copies from its dad. So it really has like 40 copies of p53. What does that enable it to do? That enables it to really make sure that if it has any cells that are beginning to transform and that should undergo apoptosis, right, it has plenty of good p53 to be able to trigger the apoptosis. Remember, we said the problem with our genomes is because we have just one copy of p53, right? But we're diploid, so we actually have one from mom and one from dad. If you inherited one bad allele, or you had one of the p53s from mom or dad, right, ends up getting hit, right? That causes a big problem somatically in the cell because p53 functions as a protein tetramer. So if you have a point mutation in one of the alleles that causes the protein, the p53 protein that's made from that one, to not be able to bind to DNA correctly, that messes up the ability of the tetramer of p53s from being able to do it, right? So only 1 out of 16 possible configurations of p53 monomers, right, would actually be a good wild type p53. The other 15 would have, some bum version of the p53 as part of the tetramer that would mess up its function, right? So the elephant, though, by having 20 copies of p53, it's able to make sure that that problem is not nearly as big of an issue as it is in humans, right? So amazing to think that this is the way that elephants have evolved to be able to cope with their immense body size and immense risk of developing transforming cells. All right, so we've studied p53 and we've studied telomere loss. Let's kind of put this together and think about the types of steps that might happen as a transforming cell experiences mutations that lead to things like improper shortening of telomeres, leading to that are not properly detected by things like p53. Let's imagine that here you have one chromosome out of a genome of a particular cell that is already starting to transform, right? So there are different mutations that have already taken place in here. Maybe you have RAS mutations, maybe you have SARC mutations, maybe you have MYC mutations that are already starting the cell to proliferate out of control, right? Normally what would happen? Normally, as the cell is dividing, right, if this is a epithelial cell, right, since we use that as our sort of model of understanding how cancer progression happens, once that differentiated cell begins to proliferate too many times, its telomeres are shortening. And so you depend upon something like p53 to detect crisis is happening, right? And then tumor suppressor genes like p53 would then put the brakes on the cell, right? And perhaps encourage it to undergo apoptosis. But what happens if, for example, you then get a p53 mutation that results in p53 loss? That means the cell, the transforming cell, can continue dividing, right? And with each division, its telomeres get shorter and shorter. It passes through crisis, right? Because without p53 available now, right, it doesn't stop, it keeps dividing. And so eventually what happens is you end up with a situation where you have those breakage fusion bridges forming. Remember, we have these sister chromatids here that have lost their telomeres, perhaps because of too much cell replication or division. They begin to fuse together. When the cell is forced to go through mitosis and the sister chromatids are pulled apart, that's when we have those random breaks that happen. Because remember, I always remind the students this is just one long molecule of DNA, right? So just because they fuse together over here doesn't mean that this particular junction point is any different than any other spot along the DNA once the DNA is together, right? So if the DNA is going to get torn apart, the breakage is probably going to happen somewhere else over here. And then you end up going through multiple rounds of this breakage fusion bridges happening until the genome gets progressively more and more and more messed up, right? And that's how you ended up with those karyotypes that we looked at, where the chromosomes look like candy canes, right? All these different colored because all these different regions of chromosomes have become fused together as part of repeatedly fusing, torn apart, fusing, torn apart. But the key thing to keep in mind is that whole out of control, unstable, right, series of breakage fusion bridges can only go on for so long. Why? Because the cancer cell, even though it's a messed up cell, it still is a living cell, right? That means it still needs to carry out certain fundamental cell functions that have to do with metabolism, membrane maintenance, things like that, right? But if you keep going through these breakage fusion bridges, eventually you're going to get to the point where the cancer cell's genome is so messed up that it cannot even survive anymore. Right? So that certainly would be the end of this kind of quest for immortality, right? If the genome gets so messed up that all the genes are no longer regulated properly and the cell can't even survive as a cell anymore, that would be the end of the road. So what happens then in order for transforming cells to be able to get through that? What happens is they have to break free of that breakage fusion bridge cycle, right? And their chromosomes have to stop sticking together. How does that happen? Because, right, in a subpopulation of these cells that are crazily going through this process, some of them, I mean, they're all getting random mutations with every cycle, right? Some of them will get mutations that reawaken TERT. So TERT up until now, because this is a fully differentiated cell in your body, the TERT genes, right, that code for the TERT complex that enable telomeres to be extended or maintained, right, those genes have been turned off as in terms of going from a stem cell to a fully differentiated cell, right? And so your fully differentiated cells normally don't express telomerase, and that's why the telomeres are allowed to get shorter and shorter as part of keeping a tally of cell division. But these crazy transformed cells with the really messed up genomes, some of them may accidentally stumble upon mutations that wake up the expression of TERT again. And so what happens is you then start having the regeneration of telomeres, right? And so after multiple rounds of crazy BFB, giving you your crazy candy cane looking karyotype, right? Eventually that has to stop. If TERT is reawoken, then telomeres are restored, and then that messed up genome is stabilized, right, in its horribly messed up state, right? And that's why you end up with cancer cells that can then continue surviving. Right? Okay. So as time goes by, even in normal healthy cells of our body, our stem cells that are sticking around and constantly dividing to repopulate the fully differentiated cells, right? They are also accruing things like mutations that just happen from the course of, let's say, being exposed to certain mutagenic compounds, exposed to environmental factors like UV radiation, or just by a function of the fact that every time a stem cell divides, it needs to replicate its entire genome, and mistakes happen, right? And any mistakes that happen in the stem cell that affect its genome, from that moment on, right, that genome becomes the new template for all future differentiated cells that are going to be made from that stem cell, right, to carry this baggage. So as complex organisms become more and more complex, right, there is this big issue of balancing these different considerations. On the one hand, you want to make sure that differentiated cells don't stick around too long in your body, because they're the ones that are more subjected to getting things like mutations, right? Because they're either more exposed to UV radiation, or if these are epithelial cells that line things like our GI tract, right? They're exposed to all kinds of compounds that you're eating as part of your food. So you don't want your differentiated cells to stick around. So that means you depend upon your stem cells to constantly replenish them, right? And that way you fight cancer because you prevent cells from sticking around too long. But on the other hand, because of this overdependence upon your stem cells to constantly replenish your differentiated cells, as your life goes on, the quality of your stem cells get worse and worse and worse, and so does the differentiated cells that are made from them. So therefore, our existence, right, our human evolution and how we are designed is a balance between the damaging effects of age, right, weighed against the damaging effects of allowing cancer to take place. So it's this careful balance. So short telomeres like we have, and also turning off the telomerase like we do as our stem cells begin to differentiate, right? It's an evolutionary trade-off between preventing cancer while at the same time accepting age-related damage. What do I mean by that? This, I think, is a really interesting kind of thought exercise. This was an article that I got out of, it was a Canadian, I think, newspaper, right? Well, actually, it was a journal. I guess it says Journal of Population Research. Right? So from a few years ago. But it was a really interesting look at how human life expectancy has changed over the years. It starts off with 30,000 years ago. This is when the last time when Homo sapiens and Neanderthals coexisted, right? And there was actually interbreeding between Homo sapiens and Neanderthals. But then from about 30,000 years forward, that's when the Neanderthals all died out and Homo sapiens, us, right? We were the last of these, hominids to continue on. So at that time, 30,000 years ago, the typical life expectancy was about 30 years, right? So that's pretty good. That meant if you live to 30, that's pretty good, right? I like this comment here. The main causes of death were, they died of injuries caused by rock falls, right? That's kind of sad, right? Because these days you don't really worry about dying in a rock fall, unless you drive over the Pali every day like I do. But anyhow, or hunting accidents, conflicts, food was very scarce, so it led to a lot of malnutrition. When you had groups of hunter-gatherers, then you would end up spreading diseases, all kinds of nasty things from animals like rabies, tuberculosis. It's pretty harsh back then, 30,000 years ago. So if we fast forward to about 8,500 BC to 3,500 BC, so this is our Neolithic period. So then the age expectancy, they now estimate, was probably around 38. Okay, that's because of the advent of agriculture, being able to start growing our own food, right? But at the same time, the more agriculture, it meant more populations were coming together in sort of more urbanized areas, and that, again, contributed to the spread of disease. Fast forward to 500 BC to about 500 AD. This is the classical Greek and Roman era. The life expectancy was 35 years old because now you had real dense populations in cities, right? You think about ancient Greece and Rome, and that meant diseases like tuberculosis, typhoid fever, smallpox would spread like wildfire through populations, right? During the medieval period, 580 to 1500, you actually have a bump in life expectancy, right? Due again to sort of, more established agriculture. But then by the late medieval period, you had these plagues, right? Like the Black Death, bubonic plague, things like that, that just decimated millions and millions, right, in the population in Europe and Asia. About around 1500 AD, if you lived to 38, that was considered pretty good. Right? Then you jump up to the 1800s. So from the 1850s to 1900, this is the Victorian period. Now you have age expectancy is about 40, right? And then finally, if we move into the 1900s, you have better health care now, right? Medicine, sanitation was a huge thing, the improvement of things like actually having plumbing and things like that, right? And so our life expectancy shot up to 70 for men and 75 to women. And now these days, right, as a continuation of that, our life expectancy is about 82 years for men and 85 for women, right? Ironically, though, notice this in modern day, right, times is when we worry about things like cancer and heart disease and stroke, right? Those have become some of the biggest killers, right, in our modern society. So it's strange to think that things like cancer really weren't a big concern, back in, let's say, earlier times, right? When people lived to be about 40, and certainly times when people only lived to 35 or 30, right? So let's say when you look back upon this history here, you can get a sense that Mother Nature intended our typical lifespan to be something like what, 35 to 40, right? And everything beyond that is really just a function of our technology, better healthcare, medicines, sanitation, the technology to control our immediate environments, right? To keep us safe from exposures of the environment, keep us safe from exposures to predation and things like that, right? But in reality, if that's only happened, let's say from, I don't know, the turn of the 1900s to today, I mean, that's like no time at all, right? That our life expectancy has gone from 40 to like 82 or 85, right? So what does that mean in terms of how we are designed evolutionarily, right? It means that we're really only evolutionarily designed to live to about 35 or 40, right? So that's an interesting thing to think about. And the fact that we live to 80 or longer, that's why cancer becomes an issue. Okay, so this idea of age, right? I already touched upon this earlier in this lecture when I mentioned how a mouse that only lives maybe 3 years, right? And that's how it's evolutionarily designed to live 3 years, right? It doesn't care about cancer nearly to the extent of something like we do, where we live decades, right? But even then, when I say decades, right, if we believe that Mother Nature designed us to live maybe 4 decades, right, 40 years old, the fact that we live double that pretty typically now, right, means that we are now having a much longer time where the baggage of mutations that we accrue in our tissues can do damage. So let's consider this. Again, our body is designed to make a choice between preventing cancer while at the same time having to accept that the lesser of two evils is we accept the damaging effects of age, right? Because we all grow up as little kids seeing old people around us, we never stop and think, why do old people look old, right? Why do their bodies fail? After all, if stem cells are present in our body that continuously regenerate tissues, right, why do we end up, right, frail and wrinkly, right, when we're in our 80s or older? Again, this has to do with the fact that Mother Nature never really intended us to live that long, right? Mother Nature designed us to have a robust system of regenerating our tissues for the first few decades, right? Maybe up until age 40. And then anything beyond 40 is like going beyond your warranty, right? So fully differentiated cells stop dividing, right? We're designed that way to prevent transformation from taking place. And we have relatively few stem cells compared to the number of differentiated cells that constantly must replenish our tissues. And so as we age, right, our stem cells pick up baggage in the form of mutations. And as I said before, that means once a stem cell gets a new mutation and that's part of its genome, as it's doing its job of repopulating all the differentiated cells, from that moment on, all the differentiated cells that it can make will be carrying that mutation with it, right? So over many decades of life, the buildup of mutations means, for example, the proliferative potential of the stem cell might be negatively impacted, right? It's not as good anymore at dividing. And importantly, all the differentiated cells that it makes, the quality of those cells are not nearly as good as the quality of those cells decades before. Okay? We looked at this figure again, but I think this is the best figure to look at this. When we said you look at the skin of an 18-year-old versus the skin of a 76-year-old, right? There are stem cells down here that are constantly replenishing the differentiated cells, but the quality of the differentiated cells, the keratinocytes that are made by the stem cells in the 76-year-old, right, are not nearly as good in quality as the teenager's skin cells, right? Both because the stem cells in the 76-year-old have lost some of their proliferative potential, so they simply don't make as many keratinocytes anymore, and whatever keratinocytes they make are filled with mutations that have naturally built up in these stem cells, right? So that's an interesting consideration, right? It's a trade-off. This is what I mean when I say that we evolved to trade off. We are so hyper vigilant about preventing cancer from happening, right? Because there's such a high potential for cancer to happen because we live many years and we're made up of so many cells. So we're so hyper vigilant about preventing cancer that the way we do it is we accept the fact that our telomeres have to shorten and we can't allow differentiated cells to stick around. So we have to constantly depend upon these aging stem cells to reproduce all of our differentiated cells, right? And that means we accept the downside of aging, right, as the price that we pay for being so good at fighting cancer. So this is where you can think about this whole lifespan thing coming into play here. Let's say you have somewhere in the body this stem cell, right? And this stem cell is in charge of proliferating and replenishing populations of differentiated cells, which are represented over here, right? That make up our normal tissues. Along the way, as the stem cell divides, right, some of those daughter cells through these what we call transit amplifying generations of cells, right? So as the stem cell is dividing, it's differentiating more and more and more as it creates more and more differentiated cells down at the bottom here, right? Along the way, some of them are going to get random mutations, but it's okay, right? Because those cells should take care of themselves. They should do the right thing, right? If there are damage that cannot be replaced or repaired, they should undergo apoptosis, right? So P53 should do its thing, taking those cells out of the picture. Let's say, though, that in the course of these cells dividing, you have this one cell over here. Let's say that it starts getting particularly aggressive oncogenic hits, right? And so it's now this red cell. So again, as it divides, right, hopefully the daughter cells can detect that there's something wrong and eliminate it, right? But maybe you have some daughter cells that make their way through. And eventually those daughter cells start getting other hits, right? So to the point where they start generating the formation of a tumor, right? The start of a benign tumor. Right? If this is a mouse, maybe that whole process, right, doesn't really reach this point until after 2 or 3 years, right? 2 or 3 years of this red cell undergoing division and getting more and more mutations, starting to form a benign tumor. But guess what? That's the end of the lifespan of the mouse anyhow, right? So that's why in the mouse, it's not worth it for it to evolve super crazy sort of anti-cancer steps, like having things like short telomeres, right? Because by the time it develops things like benign tumors, right, it's at the end of its life anyhow. Right? So there's no incentive for really good genes to be able to necessarily detect these tumors and fight them. Right? But in our bodies, right, this slide keeps going. So if it turned out that this stem cell at the top was part of one of our tissues, right, and you have this red cell forming over here, right? It has many, many more years of life to build up more and more and more transforming tumors, right? Because we live like 30 years longer than the mouse. Right? And that's why we needed to evolve better mechanisms for keeping tabs on our differentiated cells, like having shorter telomeres. Right? So that's why age is the single greatest risk factor for cancer, right? If you were to ask somebody off the street, hey, what's the biggest risk factor for cancer? You probably get things like, oh, smoking, right? Or, oh, obesity or something like that, right? But that's just because that the single greatest risk factor, age, right, is so just inherently part of who we are and our life that we don't really think of it as a risk factor, but it is. The longer we stick around, the risk of us developing cancer in our body's tissues shoots up. Look at this here. Right? Here's comparing the rates per 100,000 men of different cancers and the rates per 100,000 women of different cancers. All right, so for example, the yellow line is prostate cancer, then you have lung cancer, pancreatic cancer, colorectal cancer. Right? Keep in mind that the y-axes here are different. So for example, on the men's side, it goes up to 1,200 per 100,000. Right? On the women's side, the data just tops off at 500 here. So in reality, even though it looks like, for example, the blue line is colorectal cancer, you might say like, wow, I had no idea that women have so much higher colorectal cancer rates than men. They don't, right? Because once you actually compress this axis down, the 500 would be like over here somewhere. So it's actually somewhat comparable, right? But the important thing is that look at where the incidences or the rates of cancer really take off. They take off between ages 35 and 45, right? And after 45, that's when the rates of different cancers go shooting up in both men and women. Why is that significant? Because what did we talk about earlier this lecture about what Mother Nature intended our life expectancy to be, right? Mother Nature intended our life expectancy and therefore designed our bodies and tissues, right, to be prepared to live 35 to 40 years. Anything beyond that is beyond the warranty. And when I say warranty, what am I talking about? I mean, like literally warranty, right? When you buy like a new flat screen TV from like Best Buy or Amazon, right, it usually comes with a warranty, like a 90-day manufacturer's defect warranty. So they figure in the first 90 days, if certain electrical components are bad, then they would have already shown up as being bad, right? Beyond 90 days, you're sort of on your own. Right? It used to be like a nice TV that you bought could last like 20 years, right? And some of you maybe still have an old TV in your family's house that's like 20 years old, right? But these days, the TVs that we buy, they're pretty crappy, right? In terms of the quality of the electrical components. They're meant to last like, 2 to 3 years, because by then you've gone from 4K to 8K to whatever, right? And they expect you to just kind of like throw away your TV and buy a new one. So as a result, the quality of the different components that are used in the circuit boards or whatever are much lower quality in order to keep the price down, right? Manufacturers like Sony or Panasonic, whatever, Samsung, they're not going to use a particular capacitor or resistor, right, that costs 10 times more than a cheaper one just because the one that's more expensive will last longer. No, if the life expectancy, the consumer expectancy of the TV is 2 to 3 years, right, they're just going to use cheap parts to get you 2 to 3 years of use. Right? Think about that in terms of Mother Nature and how we evolved to be designed this way after hundreds of millions of years of evolution, right, up until basically the 1800s or the early 1900s, our life expectancy was still, right, like 40 years or so. That's what Mother Nature intended for us. Now that we live beyond 40 years, that's why you see these colored curves, right? Because the components, right? And I'm not talking about capacitors and resistors. What am I talking about? I'm talking about our genes, right? Are designed and chosen by evolution, right, to last a good 40 years. Right? And beyond that, right? Their ability to maintain and protect against cancer, you're on your own, right? You're out of warranty. But you might say, well, but that doesn't make sense though, because why doesn't evolution favor selection, natural selection for genes that might have been, for example, really good at reversing the effects of cancer? Because wouldn't that be, right, really important? Wouldn't that be selected for if you actually have certain alleles for certain genes that are really good at detecting, for example, metastatic disease and reversing metastatic cancer, right? Wouldn't that be a very favorable something that natural selection would want to pass on? Because after all, right, our bodies have very amazing restorative powers. When we get a cut, we fully expect that our cuts can heal. When we break a bone, right, we fully expect that with a little nursing and care and listening to what the doctor says, right, our bones will heal. And we recover from things like infections very well, right? We have very strong immune systems that are constantly fighting off different types of threats, both familiar threats and novel threats. So why can't we reverse the process of tumor formation? Why is there not natural selection for genes, right, that can reverse the process of transformation and reverse cancer? Again, it has to do with this, right? When we talked about the little mouse, we said the genes that the mouse come equipped for are equipped to last it, 3 years or so, right? And that's really all it needs because there's no incentive to be any natural selection for mouse genes that can, keep the mouse going for 10, 20 years, right? So this idea of what genes get selected for through natural selection, this is at the heart of why we do not have really good cancer reversing genes in our body. Think about the natural course of life expectancy. This would be true for men and for women, but let's use the example of a woman, all right, from 6 months of age through a childhood, adolescence, and adulthood, right? Let's say these days we can expect to live a ripe, full 90 years of age, right? And during this time, obviously, the genes that you inherited from your parents are being expressed, and these define your ability to grow and to be able to fend off things like different diseases like cancer, right? So which genes are actually selected for as part of natural selection and thus passed on to the next generation? Consider this. In modern society, right, it's very common now for women to have children after the age of 30. And in fact, more and more having children closer to the age of 40. The reality is, right, the way Mother Nature intended us, if we're only going to live to 35 or 40, we're really supposed to have kids like, when we're like 20, right? And that way, if you live for another 15 or so years, that's how you can raise your child until he or she is a teenager and then can then go off and fend for themselves, right, before you kick the bucket, right? But let's say in modern society, let's push this back to, let's say, 35, right? So that's when you have children. So where does natural selection work here? Darwinian natural selection, right? Because your ability to reach this point and have kids all has to do with your selection of genes that provide you fitness, right? I don't mean fitness like 24-hour gym, go on the treadmill fitness. I mean evolutionary fitness, genetic fitness. That means you have the traits or the phenotypes that are considered, right, good, strong phenotypes that you would then, right, those are the types of traits that would be selected for by a mate, right? Whether you're talking about, a guy or a woman, right? So therefore, if you have good genes that get you to the point of having kids, those are the genes that are the important ones that get passed on. Right? But let's think about this. Where does cancer sort of rear its ugly head in this picture? We said cancers really start after this point of age 35 to 40, right? So the development of cancer really only becomes a problem because we're living beyond what Mother Nature intended as our natural life expectancy. So at what point in our lives then would the chance of us having a good allele in some gene that would reverse cancer, when would that be selected for? That would not be important until this point in our lives, right? Then you might say, oh, if you happen to have a good allele for a gene that can somehow reverse cancer, that's when it would be selected for. But the problem is what, right? Natural selection ignores those, because natural selection considers these genes that got you to the point of passing your genome on to your kids, right? So therefore, somebody who might not have good genes that impart fitness, but by chance have really awesome genes for fighting cancer when they're 80, right? They may just not, right, be as likely to reproduce and pass those good genes that fight cancer on. Right? So again, this is just a very thought-provoking way of making us realize that because in modern society we deal with cancer so prevalently now, right? This makes us aware that cancer is only a big problem in our society because of our own success of living as long as we do, right? But unfortunately, because this success has only happened in the last, what, maybe like 200, 300 years, right? Compared to the millions of years of fine tuning the way our cells work, right? We've had no time at all to select for things that do a good job of reversing cancer, right? Okay, so let's go ahead and stop there.